So that's the image of our scanning electron microscope. Nowhere near as good as a transmission electron microscope, but still very, very useful in us understanding the external structures of a microscopic bacterium. Okay, the last microscope that we'll talk about is called the atomic force microscope. These are microscopes that are used to view specimens that are live. Remember, the transmission electron microscope and the scanning electron microscope don't allow us to view live specimens because it takes a pretty lengthy process and pretty rigorous process in order to prepare the samples for viewing underneath those microscopes. And when we do that, those samples end up dying or are killed. With atomic force microscopy, which is also abbreviated as AFM, scientists are able to see living cells at a very, very high magnification and a very good resolution under certain physiological condition. When they do this, the scientists are able to see specimens inside of a solution that has been generated in order to allow those organisms to thrive and or to live. So when you're observing, it's kind of like observing bacteria that are in pond water, just like Antoine von Leeuwenhoek did. But you're just using a more powerful microscope to do that. So you have this aqueous solution, you have your microbes inside of there, and so you're able to see microbes interacting with each other, you're able to see protists interacting with each other, and this provides us with a very good you're able to observe all of the interactions that occur between these viable organisms in real time. Which is really great because we like to see how certain cells interact with each other. And in addition to this, you also end up getting to see a three-dimensional profile or a 3D type of image when you view these microorganisms in this type of framework. So AFM microscopy is very useful in allowing us to see, you know, just kind of all the intricacies that might exist between, or the interactions that might exist between cells, as well as enhance our view further of the attributes of the cells that we're viewing. And so the AFM is very, very helpful. And if you look at the image here, it uses some pretty interesting tools to allow it to work, such as a laser or a photodiode. And the photodiode is used to provide, essentially, is pretty much the sensor that allows the microscope to detect and determine what the laser is shining its beam of light on. Okay, so the laser goes and it hits the specimen, and then that specimen is bounced back, or at least the refraction of that laser is bounced back to the photodiode or captured, and that's going to perpetuate the view, enabling us to examine the specimen underneath the microscope. So atomic force microscopes are high-powered, and they're also used primarily to help us visualize living cells using a very high magnification as well as high resolution, typically under physiological conditions. And here's our final image. Here's an image of some spores that are breaking out of a bacterium. A spore, primarily known as an endospore, can be released by a bacterium when a bacterium is living in environments where it's been deprived of nutrients. And so for us to visualize this, we can use atomic force microscopy, where you have this whole bacterial cell, which you see here, and that bacterial cell is starting to break down and starting to shrink. And as these spores are being formed, they're starting to break out of the bacterium and being released into the environment. So understanding that physiological process that's taking place in the cell is very interesting. Because these spores can do a lot of damage to a patient that may inhale them. So having knowledge of how this happens, why this is taking place, what it looks like is enhanced because we have these great tools called atomic force microscopy or microscopes. And so they're very handy dandy in helping us to understand even smaller components of potentially pathogenic bacteria. Okay, so this is going to conclude our chapter 2, viewing the microbial world. So as always, if you have any questions, please don't hesitate to contact me, or please email me anything regarding this specific lecture. And hope you guys have a good week. I'll have lecture 3 posted by Wednesday. Great, thank you.